गुड मॉर्निंग सर, कैसे हो आप?
Good morning. I am fine.
सॉरी फॉर कीपिंग यू वेटिंग। और पहले जीमेल फिर जूम और वो सब करना पड़ा। एक्चुअली थोड़ा डिले हो गया। सो सॉरी फॉर द हेडेक। Our pro vice chancellor and director Professor G.K. Asari will join in 2 minutes. So मैं 2 मिनट में आपसे फिर से कनेक्ट करता हूँ सर। 2 मिनट।
Okay.
आपने
Okay.
एज यूजुअल जल्दी ज्वाइन किया है, सो थैंक यू। आदत से
Okay.
मजबूर हम लोग। सो अब 2 मिनट दे दीजिए, मैं सर को बुला के आता हूँ, देन विल
Okay.
स्टार्ट सर।
Okay.
सर, आर यू
So,
अवेलेबल?
yeah, Professor Naveen.
यस।
Yes. Yes.
Namaskar, namaskar, namaskar.
Namaste. Namaste.
So
So,
this is Professor G.K. Asari,
yeah.
our director,
Okay.
IQSC and
So,
Pro Vice Chancellor.
so Naveen
Please.
ji, I am a microbiology teacher here, and
Okay.
additional job is a academic administration pro vice chancellor and director IQAC. And
Okay.
first time we are interacting, so thanks to our Professor Sanket.
Yes.
He is handling the all academic affairs of the microbiology department, and the
Yeah.
university is now moving towards the NAC cycle 3, and
Okay.
A plus grade we have. And as a university we are in NIRF and the NAC and THE and some type of accreditations. Industry integration is one of the best practice in the university, where 18 departments, 67 different programs, 219 faculty and 4,872 students are properly industry linked. Lot of industry interaction in form of workshop, conference, symposia we are organizing. And today in the microbiology department, where BSc and MSc in industrial microbiology with 27 PhD scholars is a small family. But if you see in the biology family, we are the 50 faculty and 400 students, and 27 biotechnology, microbiology, food technology, forensic science, and pharmacy laboratories available, which are FIST and PERS recognized. So this is the small introduction of microbiology and the biology team at Amity University, Rajasthan, sir. And today we welcome you because it is online. We cannot offer a tea to you, but
No, it's fine.
maybe in future, sir, come physically and have a tea break here. Interact physically in the laboratory where students are working. So, dear students, today we have uh, Naveen Kumar Arora ji is available with us for a different type of exposure as he is at Bhimrao Ambedkar University, President of Society of Conservation of Wildlife, Editor-in-Chief of lot many publication houses, including Springer. And it is a matter of pride because if we teach 36 hour in classroom, you may feel a monotony. But if we add some senior people like Navinji with us, and the ICMR director visited last month, sorry, in the month of November end or December 1st. So this is the modus operandi in the department to take one expert every month or maybe 1.5 month gives more diversity. So, sir, I am presenting in front of you my BSc, MSc, and PhD scholar, Professor Sanket, and my faculty members Gaurav, Geeta, and many faculties in biotechnology, chemistry, chemical engineering. This is the lecture hall, and just take a welcome clap for Neeraj ji.
Thank you. Thank you.
And we will listen, we will increase our diversity with his knowledge, wisdom, as his area is also very interesting. So without taking time, I'm giving floor to Sanket. Sanket, you also take some words.
Sure.
Yeah.
So thank you, sir, again for kindly joining us. Just to briefly introduce you to the audience here and our young faculty and young students. So I'm privileged to, in my lifetime of 45 years or something, to learn and meet so many dignitaries who have really contributed a lot, including like Prof. Naveen. So he is an example, not just an academician. He strongly believes in taking things from lab to the field, not to the market, to the field. So he wants to support and he is supporting actively agri industries, agriculture farmers, polluted sites in the state of Uttar Pradesh, where a lot of applications are possible, specifically for microbiology. He has published more than 150 papers in very good journals. He has edited more than 55 books, more than my age, more than anybody's age who are sitting here or standing here.
Wonderful.
And all his publications has garnered more than 15,000 plus citations and everything. He has been awarded so many prestigious awards from India, from state government, from government of Uttar Pradesh, from different countries. I think last week you were in Sri Lanka. He was representing India, scientists from India in Sri Lankan International Conference. So he just came back a day before yesterday from Sri Lanka, representing India on a global platform. He is not just taking care of publications and applications, apart from being a bio entrepreneur. He is also a founding member and a president of two different societies, one's for taking care of wildlife as well as one big society for environmental sustainability. So this is to briefly introduce you, sir. So we are privileged to meet you virtually, hopefully looking forward to seeing you physically also soon. And floor is yours, sir. Thank you for accepting our invite. Please go ahead, sir.
Thank you. Thank you, Professor Sanket, for introducing me in such a nice manner, I should say. And thanks to the director and all the other means administrative people involved who have invited me. Mainly, it is because of you. So I'm ready to deliver my talk. Should I begin?
Sure, sure,
Yeah,
sir.
yeah.
Go ahead,
Please
please.
begin, share the screen and you start, please.
I'm sharing this.
Sir. At the top, you have some questions. Now, it's interactive. You can ask. That is what pleasure.
Is it visible?
Yes, sir. It is visible.
Keep it on full screen,
Just
sir.
make it in full
Yeah.
screen.
Yes.
Yeah, it is visible, sir.
Yeah. So, as Professor Sanket Joshi was saying, we are working in Uttar Pradesh. I am from Lucknow, Baba Saheb Bhimrao Ambedkar University, and there is a lot of problem going on in Uttar Pradesh now regarding the soil fertility and soil degradation is happening so fast. We live in Gangetic plains. We live in Ganga basin, and Ganga basin is so important for our country. I always tell my students that Ganga Basin, Brahmaputra Basin, and the Indus Basin, these three basins, which are means part of India, integral part of our country, North India specifically, these three regions provide food and water to every third person on the planet. So you can imagine the importance of Ganga Basin. This thickly populated region starting from Uttarakhand, then UP, some part of Rajasthan also, Madhya Pradesh also, some part, then Bihar, West Bengal, Bangladesh, so thickly populated areas, and this Ganga basin, what we are seeing and observing, I will try to show you that today, and what is happening to Ganga basin, which was the most fertile region of our country, and it produced maximum amount of food, means it is most important for food security in our country. And what is happening? That is what we are doing. We are trying to restore the degraded ecosystem, the Ganga ecosystem, which is getting degraded very quickly. So, see, population growth is still happening very fast, and developing countries uh, are still the population is going up in developing countries, although developed countries have stabilized or even going down. But in developing countries, including India, the population is still growing up. And to feed this, the sustainable development goals, all of you students, sustainable development goals were declared by United Nations in 2015, and one of the very important sustainable development goals is food security, that is also known as zero hunger. That means every person should get enough food and calories to survive and grow. So, food security for around 9.7 billion people, the population of our human population on planet Earth will be 9.7 billion, around 9.7 billion by 2050. So, to achieve the targets of food security for 9.7 billion people is a big task. You know, we have to increase the food production by almost 70%. This is what FAO says, Food and Agriculture Organization of United Nations, that we need to increase the food production by 70%, which is a big task. And that too we have to do sustainably. Food production has to be done sustainably, otherwise, we will get more land degradation, more water bodies will get degraded or get extinct. So, alternative agriculture practices where we involve sustainable practices like bioinoculants, biofertilizers, biopesticides, or nature based solutions have to be adopted very quickly, otherwise we can be in problem. So see, what are the challenges to food security? So there are several challenges we face to food security is facing right now. Natural disasters and climate change is happening very fast. See, natural disasters are increasing day by day, although I prefer to call them man-made disasters now. I do not prefer to call them natural disasters. Natural disasters we call like droughts, like floods, like landslides in the mountains, in the hills, heavy downpour, or maybe glaciers melting. These we call natural disasters. But now, due to climate change, I prefer to call them man made disasters, because the way we are increasing the carbon emissions, the way global temperature is rising, these disasters are increasing every year. You can check the data, how many landslides in Uttarakhand or Himachal Pradesh are increasing every year, how many cloud bursts are happening every year, how many people are dying due to natural disasters, not only in India but around the globe every year. It is increasing. So, this is due to climate change. Then, another factor which is affecting food security is soil salinity. So, this soil salinity is also increasing. People may not, means in India, uh, in India, people may not understand, particularly in our region, UP, Ganga Basin. I'm talking about that what is soil salinity, how it is increasing. People do not understand it very easily, but I will show you some data. I will show you some data. You will see that it is increasing not only in India, it is increasing around the globe. So soil salinity causes degradation of soil, it causes reduction in soil productivity, it causes reduction in biodiversity, and several other problems. And it impacts food security. When it is causing reduction in soil productivity, definitely it will cause reduction in food security. Then conflicts are happening, so many conflicts happening around the globe, you know, such as Russia Ukraine since last 3 or 4 years. Middle East conflict is going on. Several other countries are involved in conflicts these days, which is impacting the food security. Unplanned urbanization is another factor, and land requirement for biofuel production is another factor which can affect food security, because governments have made commitments around the globe, including Indian government. We have made commitments that we will reduce the use of fossil fuels, and you already must be knowing that we have now 20% blended petrol in our country. So from where to get this biofuel, bioethanol? From the land only. So that means we need to diversify the land for production of biofuel as well. So all these issues impact food security. However, today I will be discussing about soil salinity, mainly about soil salinity, how it is impacting, how it is degrading, how it is causing food insecurities. So see, this is the global land degradation map. So if you see around India, we have around 40 to 20 to 40% land degradation in our country. But if we continue like this, if we continue to behave like this, if you continue to means this is data from UNCCD. If we continue to work like this, what we are doing right now, in future by 2050, 95% of our land may get degraded. And by 2050, we have to increase food production, not decrease. So if our land 90 from 40 to 95% land degradation will increase, so how will we achieve the targets of food security? It will become even more difficult. So this is a very glaring issue right now. So this, you see, this data I have taken from Syngenta, a very big international company which is involved in agriculture sector. And in 2018, Syngenta's data says that 1 out of 9 people on the globe were undernourished in 2018. 1 out of 9. 1 out of 9 people didn't get enough food or calories in 2018. But you know what happened in 2024? The recent data you can check. I have checked the recent data of 2024, not yet 2025 data, not yet available. 2024 data says, Syngenta data says that 1 out of 5 people is now undernourished. So means the sustainable development goal about food security said that we have to achieve the targets by 2030. That means no one should be devoid of food and calories by 2030. Every person, every human being should get enough food and calories. But where we are heading towards? We are heading towards a bigger problem. We are going down the hill. 1 out of 9 people were undernourished in 2018, and 1 out of 5 people are undernourished in 2024. Why this happened? Because of climate change. The reasons I have already discussed. Because of climate change, because of conflicts happening around the globe, and one another thing which happened after 2018 was the COVID pandemic, which affected the food security. So COVID pandemic also came after 2018. So COVID pandemic, climate change, unplanned urbanization, conflicts happening around the globe, all these things have affected the food security further, and now 1 out of 5 people is undernourished on this planet. So the challenge is getting bigger. See, the orange dotted line is what the actual food demand is, and this orange line, straight line without dots, is what we are producing right now. So there is a big gap, which means we have to increase the food production. And people will say India is reliant. Yes, India is reliant, but in 2023, India government of India had to stop the export of wheat because of climate change. The temperature was very high and wheat production got down. In 2023 in northern India, temperature touched 40 degrees centigrade in March. So India, the wheat production got down due to climate change, and this drastic reduction in wheat production resulted in what? Government of India declared that we will not export any wheat to other countries, and because of which the food security of several countries got affected, like African countries where we provide a lot of grains like wheat or rice, and because of this their food security was affected. So it is not only about India, it is also about the other countries. And another thing is our population is also increasing and our land is getting degraded. So we need to be very careful and very much means clear about our policies are. See, this is where, see, this on the left side is how the food security, the global food security is around, say, around 30-40% people are suffering from food insecurity at the global level. In Asia also it is almost same like the world, the global situation. Almost for 35% people, 40% people are suffering from food insecurities. In Africa, around 80 to 85% people suffer from food insecurity. Huge. In North America and Europe, only 5 to 10% people suffer from food insecurity. And in Latin America, around 50 to 55% people, 50% people suffer from food insecurity. So see, this is what we are like, means we are unequal globally. If I'm talking about zero hunger here, SDG 2, we are unequal. But if you check about health sector, if you check about energy sector, if you check about any sector, means it will be inequality all around. It will be similar situation where health sector also, it will, you will see, check, if you check the data, it will be similar. If you check energy sector data, it will be similar. If you check water security data, it will be similar. So that means the world is unequal, and that inequality has resulted in what? That we are unable to achieve any of the targets of sustainable development goals which were declared by United Nations in 2015, and the goals and targets were to be achieved by 2030. If I quote UN Secretary General, you people know who is the UN Secretary General right now, Antonio Guterres, and if I quote him in 2024, in 2024, he said that we are not going to achieve any of the sustainable development goals by 2030. So we are failing as humanity, we are failing as people because we are not equal, we are not united. Another problem is Paris Agreement. Paris Agreement is also gone. In Paris Agreement, the target was to restrict the increase in global temperature to around 1.5 degrees centigrade in comparison to pre-industrial era. Now, in this case also, the UN Secretary General, I am quoting him, and he said that Paris Agreement, the targets of Paris Agreement we cannot achieve now. 1.5 degrees centigrade is also gone. Means the global average temperature has risen beyond 1.5, already touched 1.5 around, and it will be breached very soon. And we are heading towards, now our target should be to achieve 2 degrees centigrade. Means we should restrict the temperature rise below 2 degrees centigrade in comparison to pre-industrial era. So Paris Agreement gone. What United States government Trump has done? He has come out of Paris Agreement. So US has come out of Paris Agreement. They are not going to fund any. The US was the biggest funder for climate action. And he has come out of US has come out of Paris Agreement. That means they will not going to fund any single penny into climate action. So this is where we are heading after this crisis. Now you see, the two major factors which result in carbon emissions are energy and agriculture. So energy sector and agriculture are the most important. If we tackle these two, they are responsible for around 92% of carbon emissions on the globe, these two sectors. So if we can make sustainable energy and if we can go for sustainable agriculture, then we can reduce carbon emissions drastically. Already efforts are going on in the energy sector. I'm not going to discuss about energy sector. My work is on agriculture sector, so I will be discussing about the agriculture sector in half an hour, in next half an hour or so. So energy sector, like sustainable energy, you people must be knowing solar mission is going on in our country, solar energy and different types of, I was discussing about bioethanol, blending petrol, and EVs, electric energies, and all those things are going on. So I'm not going to discuss about energy sector. In agriculture, how do we move? Where do we move? So there is a silver lining. You see, biofertilizer production, I was talking about that. We have to move towards sustainable agriculture. And biofertilizers, biopesticides, bioremediation, these sectors are going to play a very important role in coming time. And silver lining is that biofertilizer sectors is improving by around 12%, compound annual growth rate of around 12%. Biopesticide sector has a compound annual growth rate of around 15%. That means it is growing. Very good growth rate of around 15% for biopesticides and around 10% growth rate for bioremediation sector. So these three sectors are growing, but still they are not growing at the space. It looks very nice that growing at 15% or 12% or say 10%, but not enough, still not enough to tackle the issues of sustainable agriculture, to tackle the problem of soil degradation. So we need to move even faster. So this is what we are doing. Now I come to what we are doing in the fields. We have adopted several villages in Uttar Pradesh, and at present I have 8 adopted villages in Uttar Pradesh by myself through a project funded by Department of Science and Technology, Ministry of Science and Technology. So this project, in what we have selected those villages which have very degraded soils, almost negligible productivity, almost zero productivity. Those villages we have selected in central Uttar Pradesh. So you see the transformation happening, what we have done. So these villages are affected by soil salinity. You know, in Ganga Basin, soil is becoming saline. Ganga Basin was the most fertile region of our country, particularly in North India. And it is getting saline day by day. The problem of soil salinity is not restricted to our country, it is a global issue. So 7% of the land area of our planet is suffering from soil salinity, and which equals to 20 times the area of France. And you know, it is, if the corrective measures are not taken, it is FAO, I am quoting FAO, that 50% of agriculture soil may become saline by 2050. Now, why these soils are becoming saline? Because of faulty irrigation practices, because of excessive use of chemicals, because of climate change. These are the three major factors why soil salinity is increasing. I gave you one example in our region in Uttar Pradesh, high evaporation rate in summers, temperature touching 40 plus in April. Two years back, it touched 40 plus in March. So now, although in March it is rare, but in April it is very, now it has become very common now. Earlier it used to touch 40 plus after 15th May, but now it touches 40 plus, very common in April, very common. So due to 40 plus temperature in April or even in late March, the temperature rises and evaporation rate increases, and people are doing this irrigation continuously through canal water. Water evaporates and left behind is the salt. So this is increasing day by day. And in other parts of the world, I will give you one example, one glaring example. In Central Asia, a whole sea has vanished, and it is known as Aral Sea. All I'm talking to this students that you can check this at Google. Just put Aral Sea disaster in Google, and you will see a whole sea disappeared in 40 years. And what is left behind is a salt desert. Another very big sea is Caspian Sea, the biggest landlocked sea on the globe. And this sea is in between Asia and Europe. And 25% of Caspian Sea has already disappeared. So you can also Google and check uh, that Caspian Sea disaster. It is happening right now. Right now. 25%. Aral Sea is almost totally disappeared. Gone. And Caspian Sea is disappearing very quickly. So what is left behind when a sea disappears? Salt desert. And this salt desert is very plays havoc, degradation of soil, loss of productivity. So every continent is now, except for Antarctica, every continent on the planet is suffering from soil salinity issues, and it is increasing day by day. In India also, you see, all the states are suffering from soil salinity except for the Himalayan states. Except for the Himalayan states of Jammu and Kashmir, Himachal Pradesh, Uttarakhand, and Northeast, all other states are now suffering from the problem of soil salinity, and it is increasing day by day. So how we classify soil salinity? It is like this. So if it is EC's electrical conductivity is less than 2, it is non-saline. If it is 2 to 4, it is slightly saline. If it is 4 to 8, it is moderately saline. If it is 8 to 16, it is very saline. And if it is more than 16 EC, it is very simple to check EC. Just take soil and make a solution of it in water and put this electrical conductivity electrode into it. It is a very simple equipment like pH meter. EC meter is there, and you can check the EC of the soil. So very simple, in 5 minutes you can check the EC of the soil. So where we are working in UP, this EC has become 8 to 16. It is very saline. So we have isolated microbes from these saline soils only, these saline habitats in central Uttar Pradesh. And we have isolated plant growth promoting rhizobacteria from these soils and check their activities. So PGPR can help plants in several ways. I will not go in detail. You can check papers of your faculty members also, your teachers, or my papers on Google Scholar or ResearchGate. And so PGPR not only playing role in plant growth promotion, they also help bioremediation, in biodegradation, in stress management, in combating climate change. These are the original pictures. These are the original pictures of the villages which we have adopted. Some of the pictures I have put here in this slide. You see what is happening. You see the complete land degradation due to soil salinity, nothing growing. Farmers, their livelihood has got affected. Their economy has got affected. I have even correlated it with several other factors, like education of their students, of their children is affected, like gender equality is affected, like food security is affected, like health security is affected, biodiversity is affected. So I have correlated soil salinity or land degradation with all these factors, the economic and social factors as well, and they all are affected due to land degradation. So people, these farmers, these villagers are moving towards cities or towns as to become laborers. They are forced to become laborers. We can call them land refugees, land degradation refugees, or climate refugees, any name we can give to them, because they are moving from villages towards towns or cities like Kanpur and Lucknow to become daily wagerers. To means they need some to how to feed their families and how to support their families. So we have isolated microbes from these very habitats, and we hold regular meeting with these villagers, discuss with them. I started this project around 5 6 years back. Since then we have a continuous rapport with these villagers, because we hold several meetings. This month also, in February, we are going to visit the villages. So we regularly visit Sardona, more than 400 families associated with us now, and these people are very uh, means they are associated with us. I know the name of the farmers, they know me very well now. So we selected these PGPRs, plant growth promoting rhizobacteria, on the basis of soil salinity, means how they are resistant, or what we can say tolerant, or to soil salinity, and showing PGP traits at high soil salinity. So zero tolerant PGPR we can call them, and we have selected them. They play a very important role. Excellent root colonizing ability. They should have capacity to produce wide range of PGP metabolites. Favor the plant to withstand biotic and biotic stresses. They can help in bioremediation and biodegradation. So these are the strains which we have selected. You see, several of them are pseudomonas, which can tolerate very high salt concentrations. Pseudomonas fluorescens, Pseudomonas entomophila, Pseudomonas putida, Pseudomonas azotoformans, Bacillus strains also, Bacillus subtilis, Pseudomonas taiwanensis, Bacillus stercoris, Rhizobium as well. We have got some very interesting strains of rhizobium. You must be knowing nitrogen fixers. Rhizobium cause nodulation in legume roots and fix nitrogen. Bergeringia is also part of the Rhizobiaceae family. So these are very interesting strains which we have collected from these soils only, these regions only, and they are salt tolerant and still showing PGP traits at very high salt salinity. So it took a lot of time to select all these strains, and I will just one interesting story I would like to add here. I would like to explain what we checked is that we in all these adopted villages we checked the state of nodulation in legume crops, and you know what, more than 70% of the crops were not showing nodulation. This is a big alarming situation, where 70 more than 70% of legume crops are not, due to maybe due to salinity, is the reason, or maybe due to loss of effective rhizobial population due to increasing soil salinity, but more than 70% of the legume crops were not showing nodulation. Very alarming, because rhizobium is a very important bacteria, soil bacteria, which helps in nitrogen fixation and less reliance on fertilizers. And legumes are used as crop in crop rotation, increase the fertility of the soil. So if the legume crop comes in problem due to lack of nodulation, then it will be a very bad situation in the coming times. So we checked several metabolites, several metabolites we checked for how they are behaving, how they are acting during soil salinity, when we increase the soil salinity in the media, in the broth, in the plates, what is happening. So, some of these metabolites were very distinct, very clear, that they play very important role in protecting the cells, not only the bacterial cells themselves, but they also help in protecting the plant from the abiotic stress of salinity. One more thing, soil salinity is directly related to water stress as well, and to pH stress as well. So if there is high soil salinity, pH of the soil will increase, it will become alkaline. So we call them sodic soils also, saline sodic soils. So we have checked the pH, it may be about 10, 11, like this, 12, of these regions. And water stress is also there due to osmotic stress of salt. So these strains, if they are working, they have to combat multiple stresses of water, of salinity, and of pH, high pH. So, what metabolites we could check and identify, those are playing very important role, and the most important was the EPS, exopolysaccharides. And you know what we checked? We got to know one when we all these papers are published in high impact journals. What we got is that most of the strains, whether they are pseudomonads, whether they are rhizobium, they are producing, or whether they are bacillus, they are producing copious amount of exopolysaccharides, huge amount of exopolysaccharides when we increase the salt concentration in the media. So how EPS can help? You see this picture, this figure. This is published. This is published in Journal of Advanced Research. This is my paper. So EPS can help in several ways. It helps in formation of microaggregates, soil microaggregates. It helps in trapping water, provide water to the plant as well as to the bacteria. It protects the plant. It helps in quorum sensing. It helps in nodulation, and it helps in acquisition of micronutrients. Multiple roles of EPS, and we were able to identify a novel EPS. Novel EPS we could identify known as melzitose, which was not earlier reported in these bacteria. And this melzitose is produced at very high salt concentration only. And it is produced across the strains, not only by Pseudomonas entomophila, it was also being produced by Pseudomonas fluorescens and other strains. So that means it played a very important role in saline conditions. Several different species of Pseudomonas we reported to produce melzitose. This I will not go in detail. You can download these papers. This is Acton some pictures of these EPS under different salt concentrations. So it changes the morphology, it changes its structure with increasing salt concentration. Now another interesting thing was that we check these strains for biocontrol as well, against the pathogens, soil pathogens, or the plant pathogens, sorry. So these, one of the prevalent plant pathogen in these soils where I showed pictures you in the villages which we are working. In these soils, the major prevalent plant pathogen is Macrophomina phaseolina. This is a very deadly phytopathogen causing charcoal rot disease in several crops. Reported more than 300 plants it causes charcoal rot. And see, this is how the plate of Macrophomina phaseolina looks like on PDA, potato dextrose agar. It can cause charcoal rot, root rot, head rot in several different crops and can affect the productivity. So the plants sometimes are facing both biotic and abiotic stress. Macrophomina phaseolina is a biotic stress, and salinity is a abiotic stress. So double whammy, double attack on the plant sometimes, and the whole, if the farmers are still planting some crops, they all sometimes get destroyed also. So farmers have lost confidence in farming, in agriculture, and as I informed earlier, most of them are shifting towards cities to become laborers because they have lost confidence, they have faced losses. See, so we selected some strains like Pseudomonas, Bacillus, which showed excellent biocontrol potential at very high salinity. You see the zone of inhibition here? Huge zone of inhibition against Macrophomina phaseolina, and against other phytopathogens also we checked like Alternaria alternata. And we selected those strains as well which showed very high biocontrol potential. And we also checked the mechanisms. Those mechanisms are published in these papers. So we also checked the mechanisms, how they controlled the pathogen, how they killed the pathogen, how they inhibited the pathogen. But we selected those strains as well, which showed very high biocontrol abilities at very high salt concentration. See, this up to 500 millimolar NaCl concentration, these strains are showing biocontrol potential very high salinity, and still showing biocontrol potential. We selected those strains as well. We purified the metabolites like pyocyanin and EPS to check the potential of biocontrol against the pathogen, showing to identify the exact metabolite which is working against the pathogen. We purified several metabolites and checked. I'm showing here the results which showed good results against the phytopathogen. I'm only showing those results here. So we identified, I'm not going into detail, we identified these metabolites and all those things. Now finally we produced these bioformulations using these selected strains which were showing very good PGP activities at high salt concentration, plant growth promoting activities. We developed bioinoculants. You see the names are in Hindi because the farmers, they know only Hindi here. They don't know English, they don't know any other language, they know Hindi. So we developed formulations, bioinoculants, and gave them Hindi names. And these farmers, they know by name. They ask me, sir, please provide us Jai Poshvardak. Sir, please आप हमें वो दे दीजिए ट्राइको रक्षक दे दीजिए हमें. They now know the names also of these microbes and their products very, so because they are doing very wonderful job, these bioinoculants. See, these we prepared in our lab, and we have not have MOUs with some industries as well. So we first study which I did, the pilot study which I did was with sunflower. So sunflower is an oil seed crop, very important oil seed crop. India, our country, we import sunflower oil from Ukraine and Russia, and both of these are fighting nowadays. So it has become a very important commodity to buy from other countries right now. So, and they suffer from in our villages, farmers sometimes they grow sunflower. So I was working with one of the farmers. This is one of the initial experiments around 6 years back, 5 6 years back. So his field was affected by Macrofauna facilina. You see the pictures here, and also due to soil salinity. So we put our formulations. First we checked in tubes through tube assay. This was the first experiment, so I had to be very methodical to check them in tubes, then in pots. I am not showing the pot experiment here. Then we went to the farmer, very small hold farmers here, in this area, in this region, in this state, Uttar Pradesh, most of the farmers are very small hold farmers. Small hold farmers means very little, very small land. So this experiment I did with one of the farmers. You see the control on the left corner above, and when we gave them the treatment of one of our bacterial strain, there was a drastic improvement in the growth and productivity of sunflower. So there was, see, some people ask me, say that this is a different field. It is not the same field which you are showing. So I went back to my photo library, tried to find out a picture where everything was visible in one shot. So here it is. You see on the back side is treated, and on the front side is controlled. So and you see all around nothing is growing. There are bushes, some wild bushes growing. Farmers are not growing anything because they are not interested in agriculture right now because of the quality of soil, because of zero productivity, almost negligible productivity, and they face losses. So there was improvement in every aspect of the plant growth, including germination, weight, dry weight, seed yield, which was the yield increased several times. Now we also checked this rhizobia. This previous experiment was with Pseudomonas. Then we checked with rhizobia. We got some novel rhizobial strains from the soil, like Rhizobium pascense, never reported from this region. Rhizobium radiobacter, and we selected them on the basis of there was one more strain, Rhizobium strain, which we got. It was a novel strain. So we selected them on the basis of the PGP characteristics. And during the course of this study, we devised a new method. During the course of this study, while we were working on all these bacteria, we developed a new method for siderophore detection. All of you who are working in agriculture for PGP traits, for Rhizobium or Pseudomonas or Bacillus, we check siderophore production for iron chelation if the bacterial strain has the capability to chelate iron from the soil. So siderophore production, classical method was by Schwinn and Nielands. Now we modified that method and made a micro titer plate method. At one go, you can test 180 strains with microliters of very small amount of media. So what this method has done, very fast, 180 strains at one go, very cheap, very small amount of media. Chromazurol S agar is a very costly media. One box, half kg box of chromazurol agar is around 4,000 rupees. More than 4,000, I think 4,500 or something like that. So very cheap this one, and less environmental pollution because less dye is entering the environment after the experiment is over. Chromazurol as dye is used in this method, and if very less amount is used, very less will go into the environment. More economical, fast, and less pollution. So this is one of the this is also published. This method you can download it from my research gate. So we checked against lettuce, very important crop, this rhizobial strains, and we got wonderful results, better than farmyard manure, better than NPK. We got the results from by inoculating, adding our bio inoculants. Then we signed MOU with Siri Bio in Hyderabad and produced one bioinoculant which is Hello Biome. Now this is a consortia based bioinoculant which is now on the shelves, on available in the market in South India specifically. In North India also we are signing MOUs with one of the companies known as Shri Ram, DCM Shri Ram. So this Hello Biome is a consortia. Earlier experiments which I showed were pure culture experiment based on pure cultures. Then what we noticed that it is better to use consortia rather than pure cultures. You know what consortia means? It is a mixture of strains. So we checked the compatibility of the strains. We added one phosphate solubilizer, one zinc solubilizer, one nitrogen fixer, one EPS producer, one biocontrol, like that. So we had a consortia of strains in our bioformulation, and this word this was a project supported by BIRAC. So you see here BIRAC is mentioned, acknowledging BIRAC for this project. This was another project, and we got the grant from BIRAC for producing this bioformulation and transferring the technology to the industry. Now this product was after 3 to 4 years of experimentation in lab. After we checked the compatibility, we checked how much phosphate solubilization was happening or nitrogen fixation or zinc solubilization was happening even at very high salt concentration. If we mix the strains, were they working together or not? Were they synergistic or were they antagonistic? All these experiments were done. Then we developed this bioformulation. So then we did this experiment on wheat, and I will take five. I have 5 to 7 minutes. Yeah.
Yes, sir. Time is there.
Okay. So then we did experiment on wheat, and this we did in all the villages. It is not a pilot scale study. This is a study with all the 8 villages which we have adopted wheat. More than 300, 400 farmers involved. So wheat experiment was done on a large scale just 2, 3 years back. See, this is the control. Almost, you see, farmers will face losses if we get things like this, which are visible in this picture. And this is after we applied the bio formulation, which is consortia based. The earlier sunflower and lettuce experiments were pure culture based experiment. This is a consortia based experiment, and we got productivity equivalent to Punjab, equivalent to any productive soil from where nothing was growing. So this is the drone picture of the same. You see, on the left side are the treated, and on the left side are the untreated. Still, at present, it is a very wonderful, nice gradient you can see if you go to these villages. So you can see some farmers joined us 6 years back, some joined us 5 years back, some joined us 4 years, some 3 years, some 2 years, some 1 year, and some have still not joined us. So there is a gradient of control to 6 years of bioremediation. So complete gradation is visible. Very interesting. Those who have joined 6 years back, how their soil is, how the productivity is. Those who are still not joined is control for us. So very interesting scenario there. Even the difference between 6-year-old and 3-year-old or 4-year-old is different. It is visible. So those who, if someone gets, like, I invite Professor Sanket Joshi sometime we can visit these villages, and I will show you the gradient is so wonderfully visible at present. So wheat was a very successful in all the villages, and the productivity was just short like this, from almost negligible. So farmers got confidence. They are now working, and working with us means they are now working with us. They have got confidence. Earlier, only 20 to 30 farmers in all these villages, only 20 to 30 farmers joined us. After a lot of canvassing, lot of explanations and everything, only 20 to 30 farmers joined us. But now more than 400, around 400 farmers are with us, who are working along with us in these aspects and doing this type of farming using bioinoculants. Their livelihood has improved. See, we help them to have kitchen gardens, or we can also call it backyard farming. They have small fields where they do wheat and rice cultivation, and we have encouraged them to grow vegetables in their backyard, in their kitchen gardens. So they become self-reliant to grow vegetables as well for their families, and some they sell into the market. See, they are selling these vegetables into the market. Nothing used to grow earlier here. Now it is whole complete transformation after biorestoration, bioremediation. Some of the progressive farmers are even producing bioinoculants in their homes. We have trained them. We have given them training several times in our university as well as in the villages. So some of them are producing them. Those who are progressive farmers, they are leading by example and producing these bioinoculants. Although the quality is but still that quality is 60%, 70%. Still it is working. It is good. So these vegetables have improved their livelihood, and we have made seed banks in the villages where we have, conserved the indigenous varieties. And one of the biggest success story in this project, another success apart from the restoration of degraded soil, another success story was the restoration, the coming back of one rice variety known as Rambhog rice, Rambhog chawal. It was almost on the verge of extinction. Farmers were not growing it. It is an indigenous variety in Kanpur region, and now they have started growing Rambhog chawal again. And Down to Earth magazine, Down to Earth, they covered this story. They covered this story of revival of Rambhog chawal, which was almost on the verge of extinction, due to land degradation. So several indigenous varieties are getting extinct due to land degradation, but we have now encouraged farmers to have seed banks in their homes. Some of those progressive farmers have these seed banks where indigenous varieties are stored. So all the parameters you see, pH improving, EC improving, soil, organic carbon, organic matter, moisture content, microbial biomass. I am not showing the data. We have also checked microbial biodiversity. but I'm not showing the data here, already taken almost 1 hour. So this is the transformation you see from completely degraded soil to brown soil. Initially, the soil was white, like powder, talcum powder. And now it is again brown and it is becoming green. I have some satellite pictures as well of the transformation. And farmers are coming back and doing farming. Their livelihood has improved by several times, and they can means support their families in a better way. Education has improved. I know some farmers who have made pakka homes. Earlier they used to live in kacha homes like made up of ये वो their agriculture ways, वो छप्पर जिसे बोलते हैं हिंदी में. Now they are making pakka homes, linter and all bricks and all those homes. And their students, I know some farmers whose students are doing B.Tech in Kanpur University. So this is the transformation this land restoration has brought. So this is the data of how we have improved livelihood. I will. This is the story of Rambog rice, which I have already explained. This is the coverage, media coverage we have got over the times. This technology has received awards from several IIT Kanpur, IIT Delhi, Unnat Bharat Abhiyan. We have also got received award from Unnat Bharat Abhiyan and several other awards. And as Professor Joshi was mentioning, just returned from Sri Lanka because they wanted to see this story only. I was the only Indian representative in that conference. This is this is one of the book series which I am doing, Microorganisms for Sustainability. We started this around 8 years back, and this is Scopus indexed, and we have already published 8 volumes. You are welcome to submit your chapters if you have book chapters, or I can invite Professor Sanket Joshi to contribute 1 volume with his colleagues in this book series, which is Scopus indexed. Then this is a journal I started around 7 or 8 years back. Environmental sustainability. It has an impact factor of 2.8 at present, and it is Scopus index as well as indexed in Web of Science. Everywhere it is indexed. So you can submit your papers in this journal as well. This theme of my research is the village task to go in these villages, because these villages are quite remote. Sometimes we take from Lucknow to Kanpur is only 2 hours, but sometimes to go deep into the villages it may take 4 or 5 hours, like that, because transportation is not that good in the villages. So thank you very much. I hope I finished in time. I complete 1 hour. Thank you.
Thank you. Thank you very much, sir. So I think all the listeners, they must have taken something like I have understood many different things. So my take-home message might be different than you. So it's very difficult to summarize this years of work in one line. But I just want to say, like, if you are investing into market or something, you have invested into Tata or Reliance, and you made some 1,000 rupees or 10% profit. You go home and your family is very happy. This is one kind of an enjoyment. But you can imagine other things also, like when you're contributing to 400 plus farmers' families and you are not making money. They are making money, they are regaining their livelihood, their kids are managing to go to good places and everything. So that is much beyond than the earning. Lakshmi is nice, but this gratitude, what you get from people. So this is in one line. So what Professor Naveen has explained was like where we are, it's quite challenging, scary at times. We are contributing to many things, apart from which we are 10 to 20% contributing to this ever-growing population of the world. We are one of the highest populous country apart from China. So what he has described is we will be in the forefront to face. खाना ही नहीं होगा तो आगे काम क्या करोगे? दिमाग कहाँ लगाओगे? So it is quite scary and challenging. But what we have seen here is it's possible. You don't need to get some technology from China or America or whatever part of the world. we are self-sufficient. We can do it. He has done it. We have some faculties here. Dr. Geeta, she is an expert on PGPR and rhizobium in Citrophos. I managed to learn few things from her while we are doing research in other part. Professor Asiri is one of the student, Akhilesh. He was also working on something like this. He did some work in Gangetic region using novel Azotobacter and rhizobium strain. Under the Unnat Bharat Abhiyan, we have adopted 5 villages. So we didn't reach to your stage. We are still in the beginning phase. We have bioreactor. We started growing some biofertilizers for them. And last year we went to those 5 villages and we gave them the biofertilizer, and they are very happy. They were quite happy in a way, like they are asking us to give it to them, to sell it, to purchase it or something, because they got very good yield. So this year we are again looking forward to doing. It might take some years for us to reach to the level where you are right now. We managed to reach 5 villages and not in direct touch, but we are looking forward to it. Some of the students who are sitting here, they belong to farmers' family. Their parents are having farms. Some students, they are having sole business or agriculture farming only. So these are the kids who are learning microbiology. We are teaching them how to produce biofertilizers, how these things works in the field and everything. So most probably some of them, they might be interested to take it ahead. So really, thank you very much. Thank you very much for your time. So I don't know if anybody is having any questions, please go ahead. Students, you are so good. They understood everything. They are taking some time to go through the images in their mind. Please come, Shilpa. Please come here. So we have one of the PhD student, Shilpa, and master students. They would like to ask some questions. Please,
Yeah, yeah.
please, Shilpa, come.
Hello, sir. So first of all, thank you for this informative presentation. I just have one small question. The consortia that you are using, is it crop specific or broadly
No,
classified to
no, 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 no.
different
No,
crops?
our target it is not crop specific. We are using it the same consortia we are using for paddy. We are using same consortia for wheat. We are using same consortia for other crops. So it is not crop specific. Actually, our aim was not exactly the crops. When we begin with, we thought that if we improve the soil quality, and soil quality has improved drastically. So if we improve the soil quality, then the crops will manage themselves. That means the productivity will definitely increase. So it is not crop specific at all.
Thank you, sir. Thank you.
Good morning, sir. So my question to you is that how does the exopolysaccharide that is secreted by the PGPR biofilm helps in enhancing the plant growth in saline soils?
I showed you one slide, I showed you one slide where the mechanisms of exopolysaccharide were visible. So that paper is published as well. I published one paper exclusively on EPS, role of EPS in improving the soil, means productivity and soil quality. So they help in aggregate formation, they help in water stress, they help in biofilm formation, they help in nodulation, they help in micronutrient acquisition, several ways. Exopolysaccharide is very important in saline conditions. That's why these strains, these bacterial strains produce a lot of exopolysaccharides. As we could see, across the species, across the genus, they were producing exopolysaccharides.
Thank you, sir.
Hello, sir. This is Dr. Geeta. I would like to ask
Hello.
at the end when you used the consortia, is there any difference in the electrical conductivity, means the salinity decreases, or it's that all these are halotolerant organisms? So they are.
No, EC is improving, It has come down to around 4 now. From
Oh.
from 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 12 to 16, it has come down to 4. The organic content, the soil organic matter has improved a lot. Soil microbial diversity has improved. Microbes are making a comeback. So this is very cheap, this technology. I forgot to mention it. Only 4,000 to 5,000 rupees per hectare. The expenditure for the farmer is. Although we have just in the at present we have given them free of cost because I was supported by DST. But if they buy from market, it is only about 4,000 to 500,000 rupees per hectare, their expenditure. And so we ask them to put it with every crop.
Yeah.
It's not like कि कई बार मुझसे फार्मर बोलते हैं कि अगली बार इसको डालना है कि नहीं. तो मैंने कहा नहीं, you have to put it with, like you put a urea or
Yeah, yeah.
other fertilizers every time when you plant some crop, when you go for some crop. So similarly, instead of urea and pesticides and other things, you put this formulation in the field.
Thank you very much, sir. It was wonderful when we see that it's really impacting the people who are, working as farmers, and it's increasing the productivity. It was really humbling to, know that, and it's really inspiring. Thank you very much.
Thank you. Thank you.
Good morning, sir.
Good
So
morning.
my question is, are bio fertilizer and नहीं biostatistical कुछ था. So are they both effective in saline environment?
What?
Biostimulants and biofertilizer, are they both equally effective in saline environment?
No, you see, we developed one. Now we are using consortia-based formulation with the Hello biome, which I showed you, has 6 strains in it, and it is effective. Now we are using this consortia-based formulation only. We are not using pure culture. Earlier, I used pure cultures. I used to use around 6-7 years back. I used pure cultures, but I could see that results with consortia are far better than the results we got from pure cultures. So we shifted to, with time, we shifted to, with our experience, with our what we got the data, the results from the fields, we shifted towards consortia. So now we are using consortia, which is giving significantly better results in comparison to the pure cultures.
Thank you, sir.
Thank you, sir. So it's like a deja vu. I'm teaching some industrial microbiology courses, fermentation and everything. So I don't know, it's fourth semester BSc or MSc, second semester. I was teaching them about isolation of pure culture, producing strain and everything. And last week or something only, I was telling them about consortium. So it's like those group of 4 students who are very good friends who are sitting with each other. ऐसा होता है ना, दोस्त होते हैं. कुछ दोस्त कंट्रीब्यूट करते हैं आपके सही या गलत चीजों में, कुछ दोस्त कंट्रीब्यूट नहीं करते, वो आपको बचाते हैं, और कुछ दोस्त न्यूट्रल होते हैं. लेकिन आप जब उनके साथ में होते हो, यू बिहेव डिफरेंटली. आपके अंदर का असली कलर निकल के आता है. इट्स अ डिफरेंट थिंग. बट योर परफॉर्मेंस इज ईदर एनहांस्ड और रिड्यूस्ड बिकॉज ऑफ बीइंग अराउंड विद सम ग्रुप ऑफ पीपल. दैट्स व्हाट ही इज टॉकिंग अबाउट. सर जो कंसोर्शिया की बात कर रहे हैं, हम सोइल में से या सी में से या कहीं से भी माइक्रोब्स को आइसोलेट करते हैं प्योर फॉर्म में. It's good to study them, how do they behave individually. But अगर performance check करना है, sometimes performance is enhanced when you are with a group of bright people. That's the philosophy we try to live in our life. That's how microbes are also living. And that's what the example he is talking about. Like all of us scientists, this is what we used to do. We used to isolate individual microbes and go to the market, unse product banao. But then what we realize, like he is saying, based on our experience, sometimes things works well when you have a group of microbes together. It's a consortia. So they may or may not be contributing with the production or something, but they enhances your capability. They enhances your, so sangati. Sangati ka asar, durgati ka bura asar. This is what is consortium. So it's like uplifting group of like-minded microbes together. So thank you very much, sir, again. I'm sure students are having more questions. Sir is easily approachable. You can just put his name in Google. You can drop an email to him, and he is always ready to reply. You can see he's helping non-microbiologists in the field and everything. So if you have any questions or any doubts, feel free to reach out at him anytime. You can ask me, and I'm sure he will respond back to you soon. He already offered you write good papers, good chapters, and he might help you in publishing papers. Now I think everybody knows the importance of publishing chapters or papers. What is Scopus? I think you know that also. Those are the indexing services. This is what he's taking care of and helping all over the world, not just in India. So thank you very much, sir. Looking forward to contributing further, collaborating together, and visiting your farms. It's pending. आपका और हमारा, जैसे असरी सर ने बोला, यहाँ पे चाय नाश्ता बाकी है, वैसे मेरा लखनऊ में आके आपसे और मैडम के साथ में डिस्कस करना है. सो आई एम लुकिंग फॉरवर्ड टू इट, सर. सो थैंक यू वेरी मच अगेन, सर. थैंक
Thank
यू
you.
वेरी मच.
Thank you. Thank you.